Yeah.
Get started. So I'll go ahead and introduce Greg. Greg's actually the first recipient in our sort of DYRK1A Super Foundation vetted grants program. And so he's going to provide an update for us on this project. Greg's molecular geneticists who works on personalized ASOs or Antisense Oligonucleotides therapeutics for genetic disorders. He's currently the Associate Director of ASO Discovery at the Foundation, oversees the discovery and the end phase of the Antisense programs. He also serves as a genetic advisor to the Access to Treatment Committee, evaluating various disease mechanisms and treatment eligibility. His research interests include understanding of the molecular events leading to DYRK1A syndrome, while actively investigating the personalized end-to-one therapeutic location for DYRK1A syndrome. And since 2022, Craig's been part of our MSAB program. So I'd like to welcome Craig and look forward to your presentation. Thanks.
Good morning, everyone. As always, I want to very much. So everyone wants to do is, I guess, it was good that I guess we needed the 4.7 variants. And then I'll just talk about some of the functional consequences of the loss of. So I think, the first two are actually the need.
Do you mind get your microphone 1 second?
What's that? To
It's not really sunny.
So, yeah. Okay. All right. So, yeah, so, what I wanted to just do is go through the DYRK1A gene and protein mutations. And kind of what I wanted to focus with is that although DYRK1A is the area is currently expanding, you have these common genes that are really well studied and understood in multifactorial. These are things like diabetes, cancer, heart disease, and then you sort of get to this all the way down to the more of what you guys use is a word called nanorepair, but DYRK1A system is multiple base space where I think all the tests we did there, and a lot of the work in DYRK1A has been surrounding those progress questions through down syndrome, which is probably more considered in the real space. So I think there's a lot of work to be done in that sufficient respect, and hopefully things like this grants the foundation for support. So what I want to start with is I think all the trust on it just after analyst talk is a lot of the time you'll get a genetic report from your genetic system. A lot of family conferences when I was an academic went to a few of them, and they were just called by the geneticist on the cemetery, which I don't know if that's the case for you. So what I've done is I've tried and explained it in that sense. So a gene basically provides a message to the protein. That message needs to be passed. They're very close. They kind of read in these three letter words. So the other extensions that define what DYRK1A is, so they can't take for that. So we have just get three questions. What that is that space single message package that might change the context of the sentence or the change. So that would cause the sentence to be the gap of it. If you have a friendship mutation, what this means is there's the change in the breeding frame, i.e., is a change that causes three letters or red to get. This might be an insertion, deletion, might be a mutation that is in friendship. So this is where the work here, developing separated, and then you just got the strange where Steve people each of them as well. So you can see that the context of the sentence is not pretty good sense protein stock. A nonsense mutation then leads to a stop. So basically what that means is halfway through the sentence or message, it's just a full stop.
You talk more closer to the computer here,
Sure. So
and you can see it.
So basically then the end of the gene message is lost. And basically again it's lost the context. And then the splice side is quite a nuanced mutation. Often you'll get this mutation and they might say, oh, it's going to affect splicing somehow, or we don't know how it affects splicing. But really what happens in splicing is it kind of excludes or includes a bit of information that's either supposed to be there or not supposed to be there. So again, in the context of the sentence, the word 8 is removed, so you don't really understand the context of what the cat is doing to the rat. And then in the context of sometimes when a splice site puts some information in, now it's the cat hates the rat. So you can see how they all change the context of the sentence and the original meaning is lost. And that's sort of what happens to these genes when they get mutated. So this is a two-scale gene map where the numbered bars above are the exons. So these are the coding or the words of the sentence that are put together. And then it encodes these critical regions. And the main part of the protein in DYRK1A, as I'm sure you'll hear a lot through these talks, is the kinase domain. It's really the engine of the protein. It has these other domains that allow it to function because it's so widespread and it basically turns on and off genes in various regions of the cell. It can shuttle in and out of the nucleus, which is a region that contains a lot of genetic information. And these other signals also help it function correctly. So what I wanted to put this up is, this is just an image of how the gene looks in, I guess, 3D, how it actually, sorry, the protein looks in 3D and its structure. But mainly what I wanted you to take away from this is anything that's really dark navy blue is highly structured. So that basically means that that protein needs to be folded that way in order to work. And as you can see, the main region that's all through is the kinase domain. So mutations that are in this domain really affect how this protein is folded. And the folding of this protein is critical to its function. So this is a publicly available database called ClinVar. It's often where clinicians will deposit your loved one's genetic information, and they are kind of classified as either unknown significance, likely pathogenic or pathogenic, or sometimes they're classified as benign. So what I wanted to do was show how widespread the mutations in DYRK1A. A lot of diseases, you might see like a single hotspot of mutations within one region, but you can see that there's mutations all along the gene. And this is sort of the breakdown of them. I don't need to go into too much detail, but I just thought I would slide there. But what I wanted to show next was the location of missense mutations. So a lot of the time, because it's only one spelling error, as Emily touched on in her previous talk, because of missense variant, you don't necessarily know what its effect on the protein is, but you can see that this orange box over here is actually what the kinase domain, these are the regions of the kinase domain. And you can see that a lot of the missense variants that are likely pathogenic or pathogenic cluster in this region. So it basically shows that any small changes in the structure or the way that the sentence is put together within this kinase domain can greatly affect its function, that probably also leads to genetic disease. So as I said, the kinase domain is the engine of the protein, and these changes are likely to cause disease. So I work in therapeutic development, and one of the main things is when you look at a disease-modifying therapeutic development, i.e. you're targeting the gene or the protein of interest, you really, really do need to understand the genetics of that disease and the molecular consequences of that variant, because if you go in there with a specific type of therapeutic modality and you've assumed a pathogenic mechanism and it's not that, you can do a lot more harm than good. So understanding these consequences is basically the key driver of, at least in my field and a lot of the disease modifying the genetic therapy space. You really do need to understand what this is. So I wanted to touch on that a little bit, and then I'm going to use a novel splice site mutation where we've got actual patient material from that patient and sort of the consequences that lead to that. So what I wanted to do is touch on, I guess, from my aspect, what we consider the molecular consequences of these various mutations. So you can have a complete loss of function, which is an autosomal recessive disease. So that basically means 2 of the copies that you have are defective. That's not DYRK1A. So DYRK1A is known as an autosomal dominant disease. So you only need 1 copy to be mutated. So you might have loss of function in an autosomal dominant disease, and that's called haploinsufficiency. So I'm sure a lot of you are aware of that term is haploinsufficiency. So that basically just means that 50% of the gene is not enough to maintain normal cellular function. For most genes, if you have a heterozygous mutation, it's somewhat tolerated because the other allele picks up the slack. But in a critical gene like DYRK1A and a number of other genes, 50% is not good enough. Then you have a gain of function or a toxic gain of function. And again, Emily touched on that maybe there might be some aspects of DYRK1A syndrome that might be gain of function. And this is more so where the protein actually gains a different function than what it's supposed to have. So I think in her graph, she showed that the DYRK1A had an overactive kinase. So everyone talks about DYRK1A being this Goldilocks gene because you need sort of the right amount, and just too much is bad, and just too little is bad. So gain of function diseases there, you probably would have reduced the amount of protein, right? So if you don't understand the context, and someone might have a gain of function and you're trying to increase the expression, you probably do a lot more damage than you would good. Then you have splicing changes, and splicing changes can lead to all of the above. So if it's a homozygous splicing change, so you have two copies, then you could lead to complete loss of function. A splicing change could lead to a heterozygous loss of function, haploinsufficiency, or a splicing change can lead to a gain of function. And splicing changes are probably one of the hardest changes to understand, because no matter how much you predict using computational science, and I'll get to that in a little second, it can be wrong. So I guess that's still the problem, I guess, with AI. And I'm sure if anybody's used AI, it spits out the wrong thing quite often. And sometimes, although these computational programs are well sort of validated, they don't necessarily mean that in the real world or in the real cell that's going to happen. And really, the type of mutation that you have will generally drive these consequences, and depending on the gene, will also drive those consequences. So bringing it back to sort of like what I do, which is basically creating personalized therapies for now rare patients. The consequence of the mutation directly dictates how we're going to treat that patient. It enables precise and specific like ASO design. And I'll just briefly touch on what they are in a second, but it's not really the purpose of this talk. As I said, it's essential for understanding safety and avoiding any harm that you want to do. Obviously, when you create a therapeutic, the main goal is do no harm. And as I said, it sort of underpins our therapeutic strategy at Enron. And without being 100% certain of the consequence, we would not treat patients. So as I said, the main thing is it's the decision point on whether therapy can work, what platform you can use, and how to use it. So for those of you who might have attended previous years, I've given a more in-depth talk about what these compounds are, but today it's just one slide. So these are short, single-stranded, chemically modified DNA-like compounds. They're designed to be complementary to the gene target, so that's why they're called the antisense. So if you think of the strand as the sense, as in that's providing, the sensible information to create the protein, this is the antisense to that. The advantage of them is they can be designed pretty rapidly, inexpensively, and are very specific to their gene target because you literally are targeting that sequence in the gene. We use three mechanisms at our foundation in terms of the philanthropic side. The two which you don't really need to get into, there for toxic gain of function, whether we want to reduce the gene in its totality, or we want to reduce the disease gene and keep the healthy gene intact. And then we use the splice modulating one, which basically changes how the gene is put together. So now I want to talk to you, I guess, about a case study that we have at the foundation, and I guess what the DYRK1A Association provided some support to that I'm very grateful for. So this is a mutation known as C1072-1G. I'm not sure exactly what everyone's mutation is, but most of the time I think probably what we would have had is a C dot, a number, and then a change or an insertion. This -1 just means it's not in the area of the gene that provides the message. It's in a region that provides support to create that message, which basically means understanding it makes it a little bit more complex because it's not within the protein itself. So, as most of you would be aware, you probably went for some form of sequencing, whole exome, whole genome. You got a genetic report which has a mutation on, and depending on who you saw, you might be left with these questions. Well, what does that actually mean? And I think at the time, maybe this was the case with that specific mutation. So it'll give you the information, but they don't characterize that exact consequence. So they won't say, well, it's going to do this to the protein. And basically what happened is initially this mutation was hypothesized to create a splicing change that would lead to a frame shift. So basically it was going to remove a little bit of the protein and then cause an early termination. This hypothesis initially led us to believe that this early termination was occurring, and I'll show you some data that this actually wasn't the case. That's what I meant by having cellular material. Now, this is going to be something I'm going to touch on a lot. Is really important, I think, to advance the field of DYRK1A. A lot of research has been done in the context of Down syndrome or in the context of using a DYRK1A inhibitor, that although it might recapitulate the fact that DYRK1A is reduced, it doesn't actually account for a genetic mutation in DYRK1A. And I think understanding different patients' mutations and what that actually means and what different variants cause. So some of the work that Emily
was doing to really characterize various different mutations, I think is how we're going to advance the field. Because without the patient material, I wouldn't have been able to realize that this mutation wasn't actually what a computer spat out the mutation. So this sort of entails a skin biopsy, and I think some of you might have provided that to Combined Brain, or it can be a blood draw because we can basically pull certain cells out of blood and turn those into stem cells. But in this instance, there was a skin biopsy. We cultured the cells. We conducted all these experiments, and through those, we sort of figured out that, as I said, the in silico predicted, or the computationally predicted outcome of this mutation is not really what's happening. So this sort of really affected how we can approach the therapeutic, because it sort of opened up an avenue that we didn't think sort of existed. So I'll get into that now. So what this actually shows is we have some neuronal samples here. We have a apparently healthy subject here, and we have a DYRK1A patient here. What this is a PCR, so I'm sure a lot of you know what that is from COVID. And it just shows here that this is the full length of the region that we wanted to amplify. So in neurons and in skin cells, the size is the same. But in the DYRK1A cells, you see there's two bands. So this basically means that one copy is showing that it's healthy and full, and then one copy is showing that the piece of genetic information is missing. So if you remember back to the analogy I gave you, this basically is the R in the sentence that is missing. We sequenced it just to confirm this by saying sequencing, which is a very simplistic method. We could show that exon 8 is missing, and I'll show you where exon 8 encodes in a second. And then we use a little bit more, a more powerful tool to actually show that here we can see the splice on the ventricles, exon 8 right here and right here. And these lines basically just mean that the gene is being read that way. And you can see that there's a jump from 7 to 9 here. So what that means is genetically that this patient is missing a complete word, if we're going to keep using that analogy, from the gene message. So exon 8 is missing. That encodes quite a large region of the kinase domain. Obviously, if a single base in the kinase domain causes disease, a whole chunk almost certainly will cause disease. So this is sort of, as I said, where most of people's mutations might lie. And this then sort of opened up the doors for a splicing ASO to be used for this case, rather than having to do a few of these different other modalities. So what we showed is that if I get technical, exon 8 is in frame, so it doesn't really share any sequences with other regions, because sometimes exons share letters. So interestingly enough, this is the amount of gene expression between two healthy controls. And you can see that the DYRK1A patient has a very similar amount of gene expression. But when we look at the protein, we have a healthy control here. So the intensity of the band is directly correlated to how much protein there is. This is just a loading control. So you can see the DYRK1A patients and the non-targeting control show quite a bit less protein than you see in the healthy control. And what we wanted to do to confirm that this was because exon 8 was missing, you can design ASOs to actually remove exons. So what we did is then from the healthy allele, we designed an ASO to remove the exon 8 from the other allele, and you can see that, yes, actually the removal of exon 8 from both alleles causes the protein to disappear. So here this shows that haploinsufficiency doesn't have to be at the gene and protein level. because in this case, the gene expression is similar, although one of the genes obviously is not making the protein. So then, now I want to talk about the consequences of this mutation, because again, there's a lot of literature out there that looks at DYRK1A in the form of Down syndrome or using kinase inhibitors. And one of the things that's a bit bipolar, if you want to call it, is DYRK1A's role in cell proliferation. So depending on the context, DYRK1A can either act as a brake or act as an accelerator. Typically, what it's supposed to do, in terms of just, I guess, its overall understanding is that it sort of is a master regulator of cell cycles that basically acts as a brake on cell division. So what it's supposed to do, especially in the context of neuronal cells, and this might be why patients have microcephaly, is that the cells that need to become neurons need to do what's called a G0 exit. So your skin cells and your hair cells, they're constantly dividing, right? So they go through this cycle G1 all the way through. Don't worry about that. But when it gets to a neuron, your mature neurons don't divide. So what they need to do is they need to be in this persistent G0 exit so that they can then mature, create really nice neurons and start to migrate. So one of the thoughts is that when you have DYRK1A haploinsufficiency is that you don't have sufficient G0 exit, and the cells continue to divide. And because there's this, I guess, dichotomy where in certain circumstances they've found that proliferation has decreased, but in certain circumstances they found that it's increased, I do really think that the duality of this data needs to be assessed per patient, per genetic variant, because the consequence of that could, again, really change how we understand the mechanism of this disease. So what happens in this specific variant is it appears to have increased proliferation in this context, right? So we have got access to both skin fibroblasts, which are fibroblasts from the skin, and then we converted those into stem cells as well, which we wanted to use as tools to differentiate them. So what we did is we looked at, I'll go that one. We looked at the proliferation rate of fibroblasts compared to a healthy control. That was actually a younger sample. So sometimes fibroblasts, they will proliferate at different rates based on the age at which you're sampling. So these are actually younger than the DYRK1A patient that we have. And it's important to note here that when we looked at this proliferation rate, we normalized and said that healthy control is 100%. And you can see here that the replicates that we did looking at proliferation was about 250% increased proliferation rate compared to the control. And what's important about this is what we did is we used this under serum starvation conditions. So when you starve cells of food, they should exit at G0 because they don't have any food to replicate. So they should either slow down or start exiting, not permanently because you can read into the cell cycle. But you can see here that even under starvation conditions, the DYRK1A cells were still highly proliferative. Then what we did is we also looked, and this data I'll show again in a second, but I just wanted to highlight the differences. So the DYRK1A samples is this, I guess, orange bar, and then the two healthy controls is blue. So what we did is we took stem cells, and then we wanted to differentiate them using some chemicals that somewhat simulate, I guess, the more natural process of differentiation in the cell body. And every time we split these cells or lifted them off the plate, we counted them, And you can see that along the differentiation, what you would expect is some proliferation to decrease because you're maturing these into neurons, but really the DYRK1A cells, if anything, increased. So this sort of shows at least this mutation that it causes DYRK1A to have an increased proliferation rate compared to controls. So then there are these papers, two nice papers, and I think Henry presented some data on this, and Jeremy presented this last year on how DYRK1A impairs neuronal differentiation. So what we wanted to do is look at this process both extremely early and then a little bit later down the differentiation protocol. So what we did was, again, we just used these two small molecules that turn on specific genes that drive stem cells to differentiate into neural progenitor cells. So there's a lot of ways that you can create NPCs. You can overexpress vectors, which is a very aggressive manner. This is a bit more gentle and allows you to probably study it over a longer period of time. So what we did is we differentiated them for 25 days and looked at two different control lines. And what we wanted to do is when you go into differentiation, you want to ensure that the cells that you have are pluripotent. So what that means is stem cell pluripotency just means that a stem cell has the ability to become any cell it wants to. If you gave it the right signaling, it could become a brain cell, a liver cell, a lung cell. So what we wanted to do is just stain for some markers to ensure that they're good. So what you should see here, it's a little bit hard with this. It looks really nice on the computer. It's a shame that the light can see it, but what you can see is there's good green and red expression here, and the blue is just staining for the number of cells. So the DYRK1A cells in healthy controls, they're both pluripotent, and that's what we want to see. So then what we did is at day 25, we stained for two early neuronal commitment markers, beta-3 tubulin and MAP2. So in non-disease cell, I guess at this stage, generally what you'd want to see is some of the tubulin in the soma, and it should be starting to extend into these little neurites, which are the little hands that connect neurons to each other. And they might be Nestin positive. I'm not going to show you the image for Nestin. I'm going to show you the image for MAP2. But in the context of DYRK1A, you should have fewer beta-3 tubulin cells, maybe weaker networks, and this kind of might hint to maybe delayed maturation. Same thing with MAP2. We probably want to see that in the soma. If we have processes, you want to see it there, and it co-localized with beta-3 tubulin. In a delayed phenotype, the MAP2 will be perinuclear, so it's just around the nucleus, and I'll explain that in a little second. There shouldn't be as many positive cells, and the little hands that are connecting each other typically might not have MAP2 there. So again, this is the data that we got over the 25-day period, and you can see DYRK1A just very proliferative at that stage. Because of the ambient light here, what I just kind of want to show you is that we have beta-3 tubulin here and MAP2 here. I have bigger images for those of you at the back just to show you, but I think what you can just appreciate just from the smaller image is that the two healthy subjects have a lot more intense green and purple color compared to the DYRK1A, which means that there's less expression of these proteins. We quantified this and we normalized to the nucleus or the soma of each cell, and you can see that beta-3 tubulin is significantly reduced compared to the two controls, as well as MAP2. So I've got a bigger image, and I guess I just highlighted these spots, right? So this is it blown up a little bit more, and these are the two healthy subjects. So as you can see, there's diffuse purple around the cell. It's not really just located right by the blue. So this is just the middle of the cell that contains all the genetic information. So what I'm highlighting here is that you can see these nice neurites forming. You wouldn't expect all of the cells to have neurites because this is only 25 days. We probably need to differentiate them for another 25 days. But you can see that it's starting to happen. And anywhere that you see white is where the purple and the green are together. So now when we look at the DYRK1A cells, you can see that the purple is not as diffuse. It's really intense around the nucleus. And there's not a lot of these connections. If there are, they're really short. They're not extending to each other, as you can see here. There's very little co-localization, and there's almost no long little neurites that you can see. And again, this is just comparing it to the other healthy subject. Mon, if those of you who attended, I believe it was in Orange County, Mon Pear gave a talk on a paper, and this sort of is what we were trying to look at from that paper. So what we wanted to look at, that's a little bit later down the differentiation pathway. What we wanted to do is look at the early events where stem cells go from a pluripotent state to start having these markers of neuronal commitment. So what we wanted to do is look at it at an earlier stage, at about a week after differentiation. Oops. Okay. So one of the markers that we looked at was PAX6. Again, this is from Martin Pierre's paper, and they showed that when you inhibit DYRK1A with a kinase inhibitor and look at it compared to controls, that they had a drastically lower PAX6 expression. So what is PAX6? Well, it's a transcriptional specification gene and protein that basically helps define neural progenitor fate. If there's issues with it, it sort of provides the instructions to become the right kind of brain cells. It's not necessarily just a marker. It also actively downstream. So it's what's called a transcription factor. So it kind of turns on a lot of other genes, but it actively drives downstream genes for neuronal commitment. Interesting, I think what I wanted to put here is that it can be critical for eye and cortical development. So obviously I know a lot of people's loved one might have some eye issues. And generally what lower PAX6 means might be an impaired neuronal progenitor specification. So what does, oops, let me go back. So it didn't show you the image. So what I wanted to show you here is that PAX6 is in red. And again, it looks a lot more pretty on a computer screen. But I think just from what you can see here is there's a lot less of the red hue that you can see these patient cells compared to the control cells. So just highlighting here, it's really intense. There are some positive cells, obviously, because DYRK1A is still there. So in some cells, we're probably going to get some nice differentiation. But what you can see is there's just a lower expression of PAX6. So what does this sort of mean for DYRK1A syndrome? I guess part of it shows that this mutation kind of recapitulates the hallmark phenotype of failure or delayed neuronal differentiation, both at the earlier and latter stages of NPCs. The increased proliferation is somewhat conflicting with published reports, but again, I think this really highlights the need for patient material and the fact that the more variants that we study, either through CRISPR models or through patient collected cells, the more we'll actually understand what true DYRK1A haploinsufficiency is all about. Because while I appreciate that the inhibition using kinase inhibitors has provided great value to the field. They're not 100% specific, so you don't know whether they might be doing other things in the cell. So I do really think actually looking at a cell that has a genetic mutation rather than a chemical reduction of DYRK1A is better, and that's just my opinion. People might have a difference of opinion, but I do feel like hence why I bolded this and underlined it. It's sort of, I really do feel that that's where we should go. And basically, just to conclude, it's, DYRK1A is a really important gene. It does a whole lot. I mainly touched on the cell cycle and the differentiation aspects. Mutations across the gene can affect the kinase, and I think that's why understanding the consequence is really important. And finally, this is among the first, I guess, patient-derived data sets that we have. So there's not a lot of data, like I said, surrounding actual patient material and what the consequences of those are. So I think it's an important step in the field. And it sort of highlights that increasing the gene and/or expression for true haploinsufficiency is really the therapeutic way that we need to go. And probably a common approach targeting the healthy gene or protein appears to be the most rational therapeutic approach. So that, thank you for your time. Happy to take any questions.
Thank you. So you kept talking about therapeutic approach. Could you, I guess, in more layman's terms, translate what that might mean for us as parents and kids with the syndrome, and if something were to come out that could potentially help in with the way how that might work for kids who are younger versus kids who are older, is there, some type of threshold that once they pass a certain age that they're may not be effective or some sort of.
Yeah, I mean, I'll preface it that I'm not a neurologist or a doctor. Obviously, I'm more of a geneticist than an MD. But what I can say, at least from what I'm allowed to talk about from my company's perspective, because we work on rare neurodevelopmental diseases a lot for other programs, is I think, yes, obviously the earlier that you probably intervene, the better. But for other diseases that are neurodevelopmental like that we treat, we have still seen benefit and gaining of skills in patients that are in their teens. I think like with any therapy, the earlier that you start, probably the more clinical benefit that you would have. So obviously, the younger the child is, the more neurogenesis, the more like brain cells they're making, right? And if you can increase the amount of DYRK1A, you'll probably make better connections. So while I think that early intervention is the best, I don't think that we can counter the fact that even I think in your 20s that you might be able to see benefit, because just based on the data that we've got from other clinical programs that are neurodevelopmental in nature, we do see benefits still. But definitely I would probably say the earlier you can treat, the better.
Let's guess the potential time frame that we potentially could see something maybe come out and AI have a role in speed of how quickly some of these things could be developed and studied.
I mean, that's a really tough thing. If you wanted one that's going to be clinical in nature, i.e. that's going to be a global upregulation, it's going to need to go through the phase 1, 2, and 3. Probably Oliver is better at the therapeutic development side than me, so you might probably answer that better than me. But that can take quite a long period of time to get a drug approved, like as in it's got a tick from the FDA to be dosed, widely. That doesn't mean that you couldn't enroll in a clinical trial when they come. I do know that there's several companies actively looking at ways to increase DYRK1A, and hopefully I don't think necessarily AI is the biggest driver of that. I think the advancement in our genetic understanding and the tools in which we can use to determine that data has gotten a lot quicker. Like we used to do a PCR in it'll take a couple of days, I can do it within a day. So it's more around the technology of genetic ASOs and stuff that will probably accelerate that. But probably anywhere from hopefully 5 to 10 years, you might see something maybe come to clinical trial. Might be sooner than that if data looks really good. But then obviously then they need to do a clinical trial for that.
Sorry, are there any studies or any thoughts on the kind of a nutritional approach that could potentially affect the expressing of because I know there was some talk about green tea or some kind of a thing that Down syndrome that actually reuse, which is the opposite of with our kids.
Yeah. Well, I don't, to be honest, I don't know, but I have gone to a lot of conferences where there's definitely a gut-brain axis that exists, right? So whether or not there's compounds out there that can increase the amount of DYRK1A, like I don't want to be, I guess, negative, but I think like no matter how much green tea someone with Down syndrome drank, I don't think it would have significantly affected that. I think from my perspective, being someone who works in genetic therapy, I think you need to plug the hole, right? You need a disease modifying treatment, i.e. you need to change the problem, is at least in my opinion. So you need something that targets the gene or the protein or if you could control the amount of expression in gene therapy, I think that would be an option. But obviously the Goldilocks nature of it. But you know, there's advancement in that field all the time. ASOs are one possibility, and just I think Oliver showed it. I'm not sure if you guys were here. There's a polymimic that could potentially increase the amount of gene expression, which would be fantastic. They showed good data with other haploinsufficient genes, so that's something that's coming up.
Those therapies that can come to market quicker, I guess those are something.
Well, the modality is clinically approved for lots of other diseases, so they probably have an easier path than if you created like this thing from scratch that no one's ever heard of. But they would still need to go through the same regulatory processes that any other drug. It's still the same clinical trial because they're a different compound.
Thank you. Yeah, I mean, I think the benefit that we have in rare disease with the FDA is it's called accelerated. The purpose really is getting drugs to patients quicker. And usually what that's done is based on data, so that you don't have to run these trials to necessarily get, you know, endpoints that you need to then get a traditional approval like you do with some other medicines. So there are accelerated pathways, but you know, what we've been Trey's talking about and what we've been working with like that's very data, and we first have to test that sort of in the lab, and before we even get it into something like patients, and even then there's money and things that have to go into getting it to proving that it's safe to put into patients and before. I mean, we're moving definitely as quickly as we can. We just started funding for this project this past year, but in terms of like where we're at, other patient groups, I think we're here and what we definitely as we can. Yeah, so the questions about parents, which would be contribute to that process, and I mean, there's definitely several ways. I mean, one is submitting data, through these other, right now we're using sort of rare X platform, but we going perhaps, but we first need data to understand like all the mutations with that group. I think what's been established here this morning is the importance of understanding mutation. So, to your point of AI, like in what Frank presented is AI first said this particular mutation leads to a frame shift, well, then actually we look at the mutation, it didn't lead to a frame shift, it led to an axon being excluded. So, AI gets to so far. What we've heard from Amelie is the importance of understanding, functional assessment of these proteins because it could be a gain of function mutation. If it is a gain of function mutation, then the therapeutic experience and increasing DYRK1A is not going to do it. Because again, we're in this sort of Goldilocks. Too much is not a good thing. In some other disease states, you can hit it with like a gene therapy that you can increase expression by 200% as a rate because that base needs something more than minimum. But in our case, like that's not going to be good. We need to get it within a specific threshold. So, but again, to go back to like what can be done, patient samples, submitting blood samples. We're going to be definitely working part of that happen. But plasma samples, data, we need all that information in order to follow the process of developing these treatments and understanding how best to utilize these treatments because in most genetic therapies there's not like a silver bullet, like one fits everybody, but you have to come at it with like a number of different strategies and in order to accomplish strategies we understand, part of precision. So
I just I've heard it a couple of times, but the probably is a very good deal. And all of our covered one of that that's reported to me data information for all of us. It takes time for this to be researched, the questionnaire, but that's like the number one. But there's a lot of things too that we should be aware. We need as a community to drive awareness of our DYRK1A. So awareness in our network, family, friends, everybody, so that's like more people understand it, more people like join because we need funding. So every time that you see like we love to like fundraise, we're covering our annual big fundraise that's what I think for DYRK1A. Help us raise awareness and funding that we can help with those stores too. And the third point, we need help. So if you have skills that you want to volunteer, come join us in the organization. We'd be happy to have you join our board, or if you don't want to join officially as a board member, we are always happy to get like volunteers to help us with something. I just have a quick question about looking at the mixed genetic testing report, and it was done like 9 years ago. Is there anything like if we go to see our geneticists because there's been so many changes and stuff, is there anything more we can do, like ask more geneticists other than working with his parity? So I don't know how much more information we could work for our specific child. Is there something we should do?
I'm not.
Other than just double electron.
Yeah.
And we have this area. But what information is on the report? Just tell me a lot. I don't know what that means. So is there like maybe actually going and asking if you explain? Because there have been changes, and in 9 years I imagine there's a lot of the genetics world. That's good work.
yeah, I think that if you do go back to your GI's department and the next semester, there may be more information that they can tell you because there's more information that's being published there's more public information like Craig mentioned ClinVar, which is a publicly available database, which basically shows like all the mutations that have been characterized and submitted. You know, and what Amelie mentioned are like variants of that's significant. So, but if you know that your variant is pathogenic, so then you know that probably is in a region that you know it's hard to get surprised. The stage probably in the kinase, which would be the kinase of that sense. But you know, I mean, there's different consequences. So it's like even if it's in the kinase domain, there's different consequences of what happens because you know that mutation it could be a missense mutation, it could be deletion, I mean, it could be a number of things like that. Definitely a functional assessment of what the mutation is and getting even more information. But you know, if you know that it's pathogenic, that like that really gives you enough information. It's that okay, this is what it is. But having those functional assessments, it's going to be like actually these two. So, but I just you know, one of the follow-up questions I had for Greg was just, you know, if you have this, you know, early impact on, you know, differentiation, then sort of later impact on differentiation. Do you even with those impacts, like with an ASO, for instance, like can you see sort of a rescue of the value that you don't include those assays?
We're doing that now. Yeah. Yeah. So hopefully that'll be the final nail in the coffin about the paper that we're going to hopefully put out for it. The one thing that I think I mean, this is getting technical now, but I guess you're a scientist, so I can say, is the cells don't like being electroporated and then start to differentiate them. So we're having a cell death issue with the treated ones that is obviously just the inherent issue with electroporation, right? So we're trying to figure that out. So what we're trying to do is just optimize the electroporation conditions. So it's a bit more gentle, but we're still getting a good amount of ASO into the cells, but not enough to, because what we've seen is they're very stressed, and when you want to differentiate NPCs, you want them to be confluent. So so basically that just means there's a lot of cells next to each other. They're taking up all the space on the surface that they're growing. What we've had is that our control cells have been very confluent, and then our DYRK1A cells have not been, and that is an intrinsic variation that you can't really use during differentiation. So we're trying to just figure that out. And I think we're nearly there, and I think just a couple more months, and then hopefully we can wrap that publication up. We have seen an increase in protein expression through an ELISA in the neurons, which was really comforting. The splice side is very weak, and there's a bit of variation in that, but I think the important thing to know is I don't think we know how much protein increase would be therapeutic. I used to work or be involved in some Duchenne muscular dystrophy ASOs, and the in vitro data and some of the clinical data went from the fact that the boys with Duchenne, which is a complete almost loss of this protein in the muscle, the ASOs only increased its expression a couple of %, and that was therapeutically beneficial. So you had boys who theoretically should be non-ambulatory by their late teens and generally dead in their mid-20s, still walking in their mid-20s, albeit with a bit of assistance. But before those therapies, I don't think anybody with a Duchenne diagnosis was walking in their mid-20s. So to that end, I don't know what the amount of clinical upregulation really is. There's a nice publication that shows this S-shaped curve of haploinsufficiency that it's quite significantly drops, and at that 50% is where you get phenotype. But obviously, if you increase it by 30%, so not double, it's sort of almost at the top of that because it's this really steep exponential drop. So I guess hopefully if we dose that, then we can figure that out. I think that one good thing is that DYRK1A can be measured in the CSF. So I guess one thing is, do you know if there's any CSF repositories of DYRK1A patients? Has anyone ever had a CSF draw?
So, I mean, I think that it's possible that there might be some with brain.
Because I'll chat to you after that, because if you can have a biomarker, which is basically a readout to tell how good or bad something is going, because DYRK1A is found in the CSF, what we can do is after therapy measure that, see if you're having this clinical benefit. A lot of the time, at least with what we do for ultra rare diseases, there's no real tangible measurement of are we having a benefit while apart from phenotypic. And sometimes it starks, like some of the benefit that we've seen has gone from someone being unable to independently walk to actually they can independently walk. So I mean that's obviously a massive clinical benefit. But the good thing with DYRK1A is that if you go and dose, you can pull out a little bit of the CSF and measure the amount of DYRK1A at baseline and see if there's any changes across, which is really good for the field because you can actually have a tangible measure of yes, it's working.
Yeah, I think to go back to your question too, I mean that's one sort of experiment that we've been defining rather than therapeutic. So, you know, if you have just one A loss and kind of which you said doesn't sort of has to be what we would call the phenotype. There's been papers done in like Angelman syndrome, for instance, like in mouse models where they looked at, you know, adding back, you know, the rescue that's like that various and what that says it's a praise. It's like the earlier that you get better. I think that goes for pretty much any disease and stage or intervention always, but you still see benefit in around the need to have. I know that did trials in England, you know, where there were more individuals now of seeing the words that they want for it. I think so. There's all the year we're kind of learning from other rare developmental disorders. That's what we see. What's you know, for research funding. Those are like things that we're trying to just establish within DYRK1A syndrome. Yeah, also in France, we started to work with good mouse models, but that's one thing we want to sort of write up from is like trying at various time points, like all these rescuing phenotypes, all indications would suggest that it's not that it would be that much different than other development disorders. It's just something like we need to just establish. Any other questions for you? If there is some sort of therapeutic, it's going to be something so unique that you have to like give your specific like expression and like gene like information and data and develop therapy specifically for that individual, or is it something that could be developed that would affect anyone who's got this condition?
I think it could be both. So for instance, like what we're doing at NLRM, this is because it's a splicing mutation, it's very different to a missense or a protein truncating mutation. In some sense that's good, in other senses it's scientifically really challenging just based on the genetics around splicing. But it opened up the door to do a personalized therapy. But I think other therapies, as Oliver said, that target just global upregulation or increase in gene and protein expression, could be some that could benefit a large majority of people. But I do think understanding the consequence, I do think you probably need to go through some form, and I think they do that in most clinical trials. They'll have like trial eligibility. They might need to ensure like that's loss of function, because as I said, if Emily data shows that you've got an overexpressed kinase and then you increase that, you might, like I said, you cause a lot more damage than this.
Okay, so I think what we're going to do now, since we've had like an adjustment in the agenda, is that we'll go ahead and break for lunch now, and then we'll have Lexi present after this afternoon. So just have a slight adjustment in the agenda without waiting time for the live. So we don't want to Alexa's presentation. So I guess we'll go ahead and just break now, and then we'll come back for the presentation at 12:45. That's good. Cool. All right. Thank you. Yes,
No, I just,
yeah, no, yeah.
the names.
I was checking the Excel hour
Yeah.